Good morning. Thank you all very much for joining us for our second virtual What's Up Doc. We're really thrilled that you are here and appreciate you making time for us this morning. For those of you who don't know me, my name is Joan Slaughter, and I'm the founder and executive director of the Morgan Adams Foundation. My husband and I started the organization in 2000, 2 years after our oldest daughter, Morgan, died from a malignant brain tumor. In the intervening years, we have spent time helping build the neuro-oncology program at Children's Hospital Colorado and being very active and involved in funding research for pediatric cancer across the country. This morning, we'd like to share with you a little bit more about the work that we do at the organization and how the scientists that we partner with come together across the country to help move forward better treatment options for kids fighting cancer. Today, we'll run through a brief program. We'll have the Belo family, one of our patient ambassador families, share with you a little bit about their daughter Danjela's journey through cancer. Her clinical doc, Jean Mulcahy-Levy, will share with you a little bit more about the actual treatment that she has been using and the science to take care of Danny. Her partner, David Solomon, will also share a little information about how he is collaborating with Jean on that treatment. We'll hear from Dr. Rajeev Vibhakar, head of the neuro-oncology program at Children's Hospital Colorado, about the importance of scientific collaboration across institutions and how Children's Colorado itself functions in a very pivotal role to provide expertise and build out those collaborative initiatives. Finally, we'll hear from our board president, Mr. John Dudasch, who is also a family ambassador for the foundation. His daughter, Avery, was a patient at Children's Colorado and lost her battle with the brain tumor. We hope that the information that we present to you today will be informative and help you understand a little bit more about both the need for increased support of research and funding for new treatment development for kids with cancer, and that you'll understand more ways that you can get involved in helping us continue this journey and support the initiatives that we push forward for our kids. With that, I would like to introduce Jennifer and Marc Belo and have them join us and share a little bit with you about what it's like and their journey with their daughter, Danjela.
Good morning.
Good morning. So our journey with it started May of 2016. It's just started with a headache that wouldn't go away, got worse. And within a couple hours, we had her in an emergency room. And then it went horribly awry from there, from a bad headache to encephalitis to a radiology tech that did a CAT scan asked me a question, said, did she fall and hit her head? And that was the first thing we knew that something was drastically wrong. And then within an hour, she was life flighted to Children's Hospital, at which point they opened her head up and took out a massive tumor, I think about 7 centimeters. Within 7 days to the hour, we were sitting back home on a couch going, what the heck was that this weekend? You know, we didn't even know what happened yet. And then a couple weeks later, we start getting test results back in that and find out we have a brain cancer patient now. She was put in a very aggressive radiation treatment first for about 6 weeks with a chemo push. Then after a short POSX, we went to 6 cycles of chemo. That was pretty devastating for her, but on the whole, she came out better. And we actually were declared in remission. So by January of 17, we were in a remission status. We knew something, it could come back. It may not. We were fine. So we had gone to 3-month and then 6-month MRIs. And then in the summer of 2018, the doctors started to see, they couldn't quite figure out what it was at first, whether it was growing, radiation, necrosis. So it was determined. They went back in, took a look, got another piece of tumor out. And we're starting to treat that. At this point, then we tried an experimental drug And it's still coincidence. We aren't sure if it caused it, but she started to have she was having seizures that no one quite knew until she had a pretty devastating one. And we spent almost 60 days in the hospital in the fall and winter of 2018. So as she was recovering from that, we still had more tumor growth coming. So over 2019, I think there was one growth that was we dealt with targeted radiation. Then over 2020, this past year, we went through 3 different radiation cycles, all targeted for growths that were showing back up on MRIs. Every 3 months was a new spot. So the last treatment had just ended on Christmas Eve. And by the time New Year's rolled around in the first week of January, the radiation had really taken a toll on her, where she was really not functioning at a proper level. She had a hard time even moving and getting dressed. So right now we're in some aggressive therapy. She is getting better on that aspect. But an MRI the other day, we now have a new growth. And we're looking at a longer stretch of targeted radiation. The only bright side is right now we haven't had to bring any nuclear options in of heavy chemo, massive radiation. It's just targeted. So hopefully all the symptoms and that we can sort of stay on top of and not keep lowering what she's able to do day to day now. Yeah.
Once the initial treatment was over, we thought we were okay. We thought we were in the clear and that we could start a new normal, having her recover, hopefully go on to live a somewhat normal life. But when she relapsed in 2018, we were back to square one. Every time we had a little bit of hope, genetic testing, there was no match. And with that, oh, by the way, your daughter has a very rare genetic mutation in her tumor that kind of makes it a little difficult to treat. We tried the experimental treatment, and as Mark said, that was devastating for her, gave her seizures. So we go to radiation treatment after radiation treatment after radiation treatment, and we're kind of at a point right now where what do we do when the next growth shows up? Because it's not if, it's when. We know something else is going to show up. But we're at a point right now where we're running out of options and we just don't know the answer to what's next. So it's because of the Morgan Adams Foundation and its supporters that we have hope. Hope that funding will continue for Dr. Jean and her team and all the others that are working to help find a cure and better treatment for Danny. It's needed. It's critical to continue the research and to give hope for families like us and other kids who are in this position of not knowing what's going to be next. Finally, we'd like to thank the Morgan Adams Foundation for inviting us here today to speak on behalf of our family and other families who are going through the same similar situation. We'd like to thank the supporters who continue to donate and potentially new supporters. Please, please, please do not stop giving because it is crucial to families like ours. And finally, we would like to thank Dr. Jean. You have always been honest and open with us when it came to Danny. And sorry, I tried not to cry. But you're not giving up on Danny, and you've never given up on her. And it's because of you that we have hope. And we know you have Danny's best interest at heart, and we love you for that. So thank you all again for inviting us, and that's it.
Thank you so very much, Mark and Jennifer, for sharing your journey. I know from personal experience how incredibly difficult it is to be going through what you guys are going through, what Danny's going through, and as a parent to have to watch all of it. So it's with profound gratitude to you both that we thank you for sharing your story. To talk with us a little bit about what steps can be taken to help kids like Danny, it is my also profound honor to introduce to you the docs that we work with, who are every day trying to make a difference for the kids that they're caring for and that we know. So I will hand it over to Dr. Jean Mulcahy-Levy, who is directly involved in caring for Danny.
So thank you everyone for joining us today. And just so everyone knows, Joan totally gave me permission to have a Star Wars background, which for people who know me is just appropriate for talking in this setting. So we can go ahead and go to the next slide. One of the things that we wanted to talk to you about today, as Jennifer and Mark alluded to, is as we get more information about tumors in general, we really start to learn how we can treat each individual patient based on what their tumor is. And Danny's had a very interesting journey. When she was diagnosed, like Mark said, in May of 2016. And at that time, the brain tumor organization and group in the world was going through sort of a shift and a change. We use this thing called the WHO classification of tumors of the central nervous system, which acts kind of like as a Bible for neuro-oncology. It's how we all speak the same language, so we know what kind of tumors we're talking about, and we're all talking on the same level. And in 2016, there hadn't been a change in diagnosis of brain tumors since the last publication of this in 2007. So if you go back one slide, one thing that happened in 2016 is that Danny's tumor changed title. So it had been known prior to 2016 as something called a PNET, or primitive neuroectodermal tumor. But at the time Danny was diagnosed, we were shifting from PNET to having that tumor not really exist anymore. What they had done is they had taken that tumor and looked at it in different ways genetically to find out what was causing these tumors. And all of a sudden, her PNET tumor broke into 4 different kinds of tumors and was now being called something called CNS neuroblastoma. So in a 408-page book, her tumor got 3 paragraphs. In comparison, if you look at PXAs, which are another kind of rare brain tumor, that tumor got 4 pages. So really, even in 2016, we knew very little about her tumor. Okay, next slide. But it doesn't even stop there. In 2016, as they're publishing this brand new book with what is the cutting edge data, the next cutting edge data comes out. And so in 2016, there was a publication called New Brain Tumor Entities Emerge from Molecular Classification of CNS PNETs. So taking Danny's tumor and actually looking at it in an even deeper level. And one of the things that I want to point out is that DNA methylation profiling, which is the CHIP profiling that many of our donors supported for years, really played into a lot of this kind of data. And Morgan Adams funding was really some of the first funding to get CHIP analysis done on tumors in the world. And so comparing in this international cohort, this methylation data, comparing lots of different tumor types, they were able to look at new different PNETs. So next slide. So one of the things that you can see here, this is a very complicated sort of picture. So I'm going to, next slide, go ahead and blow up one of the smaller portions. And so what they did is they had 534 tumor samples, and they looked at all of these different genes to find out different things. And one of the things that you can see is this tiny little group that I circled here, this high grade, now they're calling it neuroepithelial tumor with B core. And of the 534 tumor samples that they had, 10 of them had this particular B core mutation. And the reason that this is important is because now that we have a gene or a protein that we know is wrong in the tumor, it's possible we can target that and see what we need to do for Danny's tumor. Now, this information, go ahead, next slide, came out in Danny's tumor in 2018. Also in 2018, or around that time, Morgan Adams Foundation began funding our lab's ability to do RNA sequencing, which is sort of the next level genetic analysis of brain tumors in general. And because of that, we were able to do an even deeper dive into the genetics of Danny's tumor. And this is a really complicated sort of picture, but basically what it shows is two chromosomes that really shouldn't be next to each other, chromosome X and chromosome 22, for some reason in Danny's tumor cells have broken apart and then come back together to create this B-core fusion. So now we know not only does she have a problem in her B-core protein, we know exactly where that problem is, what the proteins that are fusing together that shouldn't be fusing together. And this gives us an even further advancement in knowledge that there's something that we might be able to target in Danny's tumor specifically because we have this data now. Go forward. So unfortunately, this is the MRI that Danny's parents were talking about. And Danny's parents and I had a meeting on Monday, and like they said, we're very frank and honest. And one of the things I want you to look at for people who aren't used to looking at MRIs is the MRI on your right with the November 2020 scan. I circled a little area and you can see the bright white, that's your fluid that floats around your brain. And just to the below that bright white fluid is this tiny, tiny white discoloration that you can see. And at the time we were doing these MRIs, like Marc said, even back in 2018, Danny's tumor is very subtle and it's very sneaky, and that it grows in such a way that it's very hard to tell immediately what's happening because it mimics what we can do to patients with treatment and doesn't only look like a tumor. And the problem with that is that it makes it very, very hard for us to tell with Danny is the problem that she's having because of the tumor or the problem that she's having because of something that we've done to her. So if you look at the scan on the left from March, what you can see is underneath that bright white fluid area, that small area of white discoloration has now greatly increased and expanded. But more concerning is that not only is it increased and expanded, it's moving across the middle of the brain. Danny's tumor has always been on the left side. And now what we see is that her tumor is not only regrowing in the spots where the tumor was, it's creeping across the midline and going over to sort of take over the other side of her brain on the other side. And we are lucky in that this tumor grows slowly enough that we'll be able to find the spots and, like Marc said, treat them with radiation. But it's exhausting for the families and it's exhausting for the doctors that in a year and a half, I have never been able to sit in a room with Danny or her parents and tell them your scan looks great. I don't see any tumor and let's hope that things continue for the next scan. For every scan for the last year and a half, we've had to have the discussion that your tumor is still growing. And the problem is that we are running out of things to do. You can only do so much radiation. Your brain can only tolerate so much. And we've tried experimental therapy, and Danny had an unfortunate seizure episode that, like Marc said, left her hospitalized and with difficulties for years to come. So now what do? We're focused on treating this new site that we have. We're focused on trying to get control of the tumor and prevent it from spreading to the right side even further. But what we really need is something not radiation, something not a standard chemotherapy, something different and new, and something that we're going to be able to treat Danny with and hopefully have it be targeted specifically against her tumor. That is what we need. And so for that reason, I have been really working to reach out to the research community in general, because, this is not something that I look at in my lab. There's not a lot of things that we don't do in research in Colorado, but B-core tumors is not one of the ones that we do. And so I went to reach out to people that I know and respect, have worked with in the past, and have an expertise in this. And so here is where we bring in Dr. David Solomon. And so Dr. David Solomon is an assistant professor in the Department of Pathology at the University of Southern California. He's also a physician scientist. He splits his time between being a diagnostic neuropathologist, meaning that he clinically diagnoses brain tumors every day. And he also has research focusing on brain tumors and sarcomas. And I went to reach out to Dr. Solomon specifically because he has an expertise in BCOR that we want to be able to provide our patients here in Colorado. So I'm going to turn it over to David so he has a chance to speak with you and share you just a little bit of information about his research.
Okay, good morning, everyone. Thanks for the opportunity to be here for the ongoing support for this new research direction that we're been working on, and for getting to hear the journey of Danjela. Hearing patient stories is certainly very motivating to us in the research field, and helps provide us the kind of motivation and courage to keep going, through all of the struggles. And research is certainly not easy. It's challenging, and hearing stories like that is what drives us in the research field. My research lab is at the University of California, San Francisco. I get to work in this beautiful place. There's a little arrow pointing at where my research lab is at the campus. This is my research lab group prior to COVID. We actually have a new lab member, but haven't been able to all be in the same place at the same time due to constraints of COVID. And here's a list of my current funding, which is basically how my research is sustained, is through the generous support of donors that have been given, as well as the National Institutes of Health. And here's my contact information and research lab website in case anybody's interested in looking more about some of the work that we do. Next slide, please. So really, there's two major new molecular technologies that are leading to rapid advances in the way that we classify and treat brain tumors and other central nervous system tumors. And these are next generation sequencing and DNA methylation profiling. And so we've already heard that both of these technologies have been applied to Danny's tumor, and this is what has been leading to the new classification and identification of these new tumor entities and new classification schemes. And so I've been heavily involved over the last year in working on the next update of that WHO classification blue book that was Jean showed. And so there'll be actually a new one coming out in the next month or two that will even have even more classification and more details, in particular about these specific BCOR tumors will be greatly expanded. And so I was one of the co-authors of that chapter and providing details. If you go to the next slide. So as mentioned, we have a dedicated focus on tumors with BCOR alterations, and have been both working on them from the clinical and pathologic description and genomics perspective, as well as making cell models. And so here are a couple of the papers that we've been involved in, studies we've been involved in. There are actually two distinct types of BCOR altered brain tumors. And the study in the top left is the one showing and describing the ones that have an internal tandem duplication within exon 15 of the BCOR gene. And these are actually the more common ones. And then shown at the bottom right are the less common ones, and these are the ones that have fusions between the EP300 gene and the BCOR gene. EP300 is a histone acetyltransferase gene, and BCOR encodes a transcriptional co-repressor protein. And so these are two genes that are involved in changing the way that genes express different transcripts and make different proteins and control the epigenetic state of the cells. And so these are the we're now interested in making cell models, if you go to the next slide, that study what do these specific BCOR alterations do. And so here is just an overview of what we do in my research lab and what it is that we're applying and doing to study tumors like Danny's. And so the first is basically trying to identify what are the genetic alterations that define the many distinct brain tumor entities. And so we're doing genomic analysis using exome sequencing and RNA sequencing, just like the results that Jean showed for Danny's tumor. Next, if you move one. Yep. And so then the next thing we do is we try and answer what are the molecular mechanisms by which these genetic alterations promote tumorigenesis. And so basically what we do is we use cultured astrocytes and we introduce or genetically modify them in a way that mimic what's going on in the tumor. And so we use new genome editing technology like CRISPR to specifically introduce B-core alterations into these cells. And then we can study them by new methods like single cell sequencing, RNA sequencing, looking at DNA methylation patterns that are actually specifically changed by these B-core alterations. And then if you go one more forward, and then what we can do is once we have these cell models is that we can identify new rational therapeutic strategies based on the pathways and genes that are disrupted by these genetic alterations. And these are through doing either drug screens that based on rational design or through kind of genome-wide synthetic lethality screens. And I think there's one more slide if you want to move forward. And so here is a method that we're using to do drug screening. So basically we're taking immortalized human astrocytes that we've used CRISPR or currently working to use CRISPR to induce this EP300 B-core fusion. We can take these genetically modified astrocytes and put them in 96-well dishes, and then we can put on them experimental drugs or shRNA libraries and do synthetic lethality screening and assay survival and identify compounds that are specifically working to kill astrocytes or gliomas that have this specific mutation or fusion. And so it's this type of research that we hope will lead to new therapies that can be used in the clinic for patients like Danny. But that's the end of what I have. But thank you very much for the support. And, I'm very hopeful that, through continued research, we will continue to make progress and be able to help patients like Danny for the better.
Thanks, David. And we really appreciate the research that you're doing. Like a lot of our donors in the past know, and maybe for people who are new to What's Up Doc, our goal is to advance the care of our patients, improve their survival, improve their lives. And when we need to, we reach out to those people that are doing the best research if it's not happening here at Colorado. So we appreciate our collaborators like David and other international collaborators that we can work with to really expand our reach. And this is research, though it's happening at UCSF, is in direct potential for improving Danny's life and coming back here to Colorado and making sure that we are curing our kids. And so I want to thank David and all of the researchers that are out there and collaborating and making sure that we really reach out, find the best research that we can, bring that back into not only our labs, but into our clinic and make sure that we're curing our patients. Danny's 16. She wants to learn to drive. She wants to, maybe work fast food. Who knows what a 16-year-old really wants to do these days? Just get out and go to school. But hopefully we can continue to treat her long enough so that David and people like him can find a cure for patients with B-core altered tumors. And until that time, we'll just have to keep hoping and searching and figuring out what we'll do. Thank you for giving us the opportunity to share you that story.
Well, thank you very, very much, Jean and David, for sharing your work and the importance of it and all of the progress that we're pushing forward to help kids like Danny. And I think the importance for our guests to understand how much collaboration makes a difference and the work that you guys are trying to push forward. So thank you very much for sharing that with us. Next up, we'd like to introduce to you Dr. Rajeev Vibhakar, who is the program lead for the program for pediatric neuro-oncology at Children's Colorado. He'll give us a little bit of an update on what's happening on our labs, in our labs here at home, and also talk about the importance of collaboration across all strains of the work that we are doing. Dr. Vibhakar, thank you for being with us this morning.
Rajeev, you're muted. You can't have a Zoom meeting and have that not happen at least once.
Okay, can you hear me now? It's like that commercial now. So thank you. And I just wanted to spend a couple minutes talking about the big picture of the Morgan Adams Pediatric Brain Tumor Research Program here in Colorado and how it fits in with all of the collaborations that we do both across the country and internationally. So this is really a snapshot of our program. And as you can see that we have this large tissue bank that was the sort of the genesis of this entire program way back in 2001 when Joan and Nick hatched this up. And really, we've been doing a lot of genomics, a lot of what you heard from Jean as well as David about the different types of genomics that we can do. You know, we started out with the array-based gene expression, which is the CHIP, which was the original large program that was being funded. And we've moved on to the RNA sequencing, and then now the single cell RNA sequencing, which allows us to actually look on a per cell basis at what goes on. And doing all of that, we identify a lot of new information, including new fusions, similar to what Danny has in her tumor. We've also got sort of three major arms. We've got one area looking at epigenetic regulation of various pediatric brain tumors, and that's similar to sort of the fusion that Danny has with the BCOR and the EP300, which is an epigenetic regulation piece. We've got a lot of immunology, immunotherapy work going on between several of our investigators, and then looking at new targets and identification, doing large scale CRISPR screens, which you just heard about with David, as well as some other high throughput drug screens. So all of our various investigators are working on all of this. And the important part really is that a lot of this information is being fed through to clinical trials. And so we've got several clinical trials that have come directly out of the programs we've done. But the important part related to all of this is that all of the collaboration that is ongoing. So, for example, looking at some of the chromatin dysregulation and epigenetic problems. Dr. Adam Green's recently actually just about to publish a big paper with collaborators at St. Jude and at Heidelberg in Germany, looking at how changes occur in tumors after we've given the patients radiation or chemotherapy, which seems to be a major problem for some of our patients. We've, doing a lot of immunology, and Nick has been working, for example, with collaborators in England, looking at macrophage activity in ependymoma. And we've been working with several other groups looking at CAR T-cell therapies for several brain tumors. We've done a lot of CRISPR screens, and for example, Dr. Venkatraman just published a paper which included 11 institutions across 5 different countries that all collaborated to drive information about a new potential therapeutic targets in DIPG, which is another devastating brain tumor. So all those bits together really help us by collaborating with people like David, with people across the country and really across the world that feed into our program and that we can feed into their programs and combine all of that information that then allows us to drive clinical trials that we then partner with other institutions to drive those clinical trials. So we have trials that we've started just here at Children's, but we also have trials that we're doing nationally through the Pediatric Brain Tumor Consortium, and then trials that are about to open through the CONNECT Consortium, which is an international clinical consortium. So by doing all of this research and all of the work that's being supported and all of the information that's being generated, we can collaborate across the world. And I should point one more thing out that has been an emphasis of our group, which is that we want to share all of the information we get openly and freely as possible. So all of our gene expression data has been submitted to internationally available expression databases that anybody can download and use. All of our single cell RNA-seq, we've created our own web portal so that we've been putting all of this information that's fully annotated to anybody who's doing any kind of brain tumor work, who wants to understand a particular gene in their favorite tumor type, can go look at that information. And because we've done all of the heavy bioinformatics lifting, they don't have to have very much knowledge about bioinformatics. They can simply go and type in, just like you would in Google, your favorite gene and your favorite tumor, and that information will pop up. So that portal is about a third of the way done, with about a third of the information in, and the rest of the information is coming as we go. We're creating the same kind of portal for all of our CRISPR and shRNA data that we've been generating, and that we literally just started about 3 weeks ago, doing the programming to create that portal, so that all of the data that we've generated in all of the brain tumors will now, so we've done the CRISPR and shRNA screens in all of the major brain tumor types, ATR, TDIPG, GBM, ependymoma, medulloblastoma. And so all of the data will now be available in publicly, so that anybody who wants to go and see what's happening in their favorite tumor type anywhere in the world can look and see what's going on. So all of those things together, I think, is really important. Important part of our pediatric brain tumor research program here is that we're able to connect with people, and we're able to take all of the information and share it as widely as possible, so that we can really drive, hopefully, therapies for our brain tumor patients faster and with more efficiency coming in the future. Thank you very much.
Thank you very much for that, Rajeev. We appreciate it. And I have to say, it has been one of the greatest pleasures to be able to work alongside the team at Children's Colorado in the neuro-oncology program and across the spectrum of the Cancer Center as they push forward the treatments that we need to really take care of our kids. So thank you. We really love working with you all. Now I'd like to introduce you all to John Dudasch, who's the president of the board of the Morgan Adams Foundation. He'll share a little bit more about the vision of the organization, his personal story, and basically the mission of what it is that we do. Thanks so much. John?
Thanks, Joan. So as Joan mentioned at the beginning of the program, I think I've got a couple of slides here that are coming up, but at the beginning of the program, the connection to the foundation is a deeply personal one for my wife and I. We're here basically because back in August of 2011, our daughter was one of the 46 kids that day that was diagnosed with cancer. And unfortunately, the science and the treatment options as they stood 10 years ago were not able to cure Avery. But I'll tell you, in listening to Jean and to David and Rajeev, it's hard not to wonder if the outcome might not be different if she was in treatment today. And that's really why, if you boil it all down, that's exactly why the Morgan Adams Foundation exists, and why we're pushing so hard for Danny and for every other kid and young adult like her. The Morgan Adams Foundation is laser focused on working with research institutions, both locally and nationally, to develop proof of concept level science. And the reason why that is so critical is the seed grants that we provide are really a critical prerequisite to obtaining much larger transformative funding grants that are necessary to truly accelerate the development of treatment options. And the reality of research funding is that larger grants are just not available without the preliminary results obtained from the seed grants, from that proof of concept level science. So that's what that is the singular purpose of the foundation, and that's why we're laser focused on providing those seed grants across research institutions. And the amazing progress that Rajeev presented is a direct result of the larger grants that the lab at Children's Hospital Colorado and the labs and David's lab have been able to secure as a direct successor to the seed grants from the Morgan Adams Foundation. But let's make this a little more granular. The advancements in science and the treatment options that in particular Jean and Dr. Solomon discussed for Danny are really why the Morgan Adams Foundation exists. We build bridges amongst researchers and act as sort of a connective tissue to increase the chances of cures being developed and deployed for our kids with cancer. We play a very active role in helping to facilitate the collaboration that Jean and Rajeev discussed to ensure that our local kids in Colorado have access to the greatest care possible. And frankly, we exist in the memory of Morgan and in the memory of Avery and in every one of our kiddos that we've lost. But more importantly, we really exist for Mark and for Jennifer and for Danny. And not only to give them hope, but most importantly, to give Danny an actual shot at life without cancer. But we couldn't do all of this without you. So we ask that you can do anything to help us help the 46 kids today, and unfortunately the 46 kids tomorrow and the 46 kids the next day that will receive the dreaded diagnosis, your child has cancer, so that they have the absolute best shot at a cure that we can possibly give them. So thank you for your time this morning. Thank you for listening to our stories. And as always, thank you so very much for your continued support. We appreciate it more than we can possibly articulate, and we wouldn't be here doing what we do without you. So thank you very much.
Thank you, everyone, for attending today's What's Up Doc session. Like we said, you can help make a difference in the life of kids with cancer. Please consider making a donation today and helping fund research. Thank you.